Hello, everyone. My name is Josh Jahner, and I'm a research scientist at the University of Nevada, Reno. And I'm really excited to be here today to tell you a little bit about my research on the genetics of bighorn sheep in Nevada. I'd just like to start off by thanking the DBC for allowing me to present my research today, and also to thank my co authors, including Mike Cox at the Nevada Department of Wildlife and Marjorie Matoke and Tom Parchman at the University of Nevada, Reno. Now, today I'm mostly going to be thinking about translocations. And translocations are one of the most important tools that we have for managing wild populations around the world. Every year, tens of thousands of individuals are moved from one location to another in order to reintroduce organisms to their previously occupied habitat or to augment existing population levels. There's a lot of different benefits from translocations, but there also, it's worth noting, a lot of potential negative consequences of translocations as well. And some of these are genetic consequences that I think about a lot. And these can include the erosion of population genetic structure among different populations, or at a bigger scale, this can result in hybridization between different lineages or different species, especially if these new populations are in close proximity with others that have different ancestry. And also, these new populations can have lower genetic diversity because they're often founded with only a few individuals compared to the source populations. So all of these different things need to be thought about, um, hopefully prior to translocation occurs. Now, one of the most noteworthy examples of a translocation success story is the history of bighorn sheep in Western North America. Everyone's probably seen this figure many, many times before, but just as a reminder, bighorn sheep used to be fairly widespread across Western North America prior to European establishment. Following that establishment, populations and range sizes dramatically decreased to it because of a number of different ranges, and the range was dramatically reduced in the mid 1900s. Thankfully, we've been able to use translocations to reintroduce sheep back onto their uh, historic native ranges and to augment populations. And it's really been one of the most noteworthy and successful translocation success stories in the history of North America. And so I'm going to think about uh, the genetics of the translocation and think about what's happened from a genetic perspective in Nevada. And so, thinking about Nevada specifically, luckily we know a lot about what the distribution was supposed to be like. And it's thought that bighorn sheep occupied nearly all of the more than 300 mountain ranges found in Nevada. And John Muir spent a lot of time in Nevada, and he said, throughout the vast region bounded on the east by the Wasatch Mountains and on the west by the Sierra, there are more than 100 subordinate ranges and mountain groups, trending north and south, range beyond range, with summits rising from 8 to 12,000 feet above the level of the sea, probably all of which, according to my own observations, is or has been inhabited by this species. And so you'll see on the left here a map of what the existing um, distribution of bighorn sheep was after that in the mid 1900s prior to translocations. So bighorn sheep used to be found throughout the state. Then when there was a dramatic range contraction. And then, thankfully, through the efforts of uh, Nevada Department of Wildlife, uh, the current distribution looks a lot more optimistic. And so the Nevada Department of Wildlife translocated in three different subspecies into Nevada. This is guided by Cohen and Hall's hypothesis of historic distributions. And basically, California bighorn were moved into the northwestern part of the state. Rocky Mountain bighorn were moved into the northeastern part of the state. And desert bighorn were moved into the central part of the state typically from those southern remnant herds that you saw on the last slide. Now, of course, all of this was done before, genetic technologies were really readily available. And so we can still go back and learn a lot about what worked well and what we might improve on in future translocations by attempting a genetic study on these populations. And that's what I'm going to talk about today. And specifically, I wanted to ask about the genetic consequences of these translocations, and specifically thinking about whether or not the three different lineages that were moved into Nevada are still genetically differentiated from one another. Also, I wanted to ask if there were any herds with hybrid ancestry, with the formation of hybrids in them. And also, I wanted to compare genetic diversity among the different lineages to see if there's any differences going there. And so I'm not going to talk a whole lot today about the methods that went into making the genetic data set. I'll let you know a few things. One is that we sequenced about roughly 1,600 individuals using a genotyping by sequencing or dd-rad-seq approach, if you're familiar with genetic protocols. And this allowed us to summarize variation at tens of thousands of SNPs. I think most of the analyses that you'll see today are based on around 29,000 loci or SNPs. And these are SNPs are just different genetic regions across the whole entire genome where there's variation, and we can use that variation to ask questions about how individual sheeps are related to one another or not. And so, moving into the first question, we first conducted a principal component analysis on those 29,000 SNP markers. And this is a plot showing the first principal component axis versus the second principal component and axis. Each point, each circle is an individual sheep that was sequenced, and they are all color coded by the three different subspecies that are recognized in Nevada. So blue is California, red is desert bighorn, and purple is Rocky Mountain bighorn. And there's two main takeaways from this slide. First off is that most individuals, most of those more than thousands of individuals cluster into really nice groups that are all the same color. And this tells us that there's strong genetic structure among those three different lineages. There's definitely California, desert, and Rocky Mountain bighorns are definitely genetically different from one another. There's no question whatsoever about it. Also, you'll notice that there are a number of purple and red dots in the center of the plot. And that's exactly where we would expect individual hybrids to be found. And since they are all both either purple or red, this tells us that there are uh, hybrids between desert bighorn sheep and Rocky Mountain bighorn sheep. And so the next question we wanted to ask is exactly where on the landscape these hybrids were occurring. Were they scattered across Nevada, or were they really localized in a couple of populations? And it turns out that the latter is true. So this is a plot showing Nevada. All the gray polygons are the hunt units in Nevada, and the white polygons are where bighorn herds are found. Each pie chart shows the proportion of the three different ancestries, potential ancestries in that herd. And you'll notice that most herds have a single ancestry. So that shows us that those herds don't have any hybrids in them. So if you have a fully red herd, every individual in there is a desert bighorn sheep. If you have a solid purple circle, every individual in that population is a Rocky Mountain bighorn sheep. Now, there's two herds that had mixed ancestry, so multiple colors in those herds. And so the first one I want to point out is the White Pines herd. It is mostly desert ancestry with some Rocky Mountain ancestry in there too. And this is actually something that Endow knew a lot about. There were four rams that um, went on a foray from the north, south, straight south. They were Rocky rams and went straight south and were found in the White Pine Range about a decade ago, a little more than a decade ago. And they were able to successfully breed with the desert bighorns there. And we can totally see that in the genetic data now. And so that made a lot of sense to us. The surprising herd is the Badlands herd. And prior to this study, the Badlands herd was just thought to be another rocky herd out on the landscape. But it appears that nearly every individual in this herd has some desert ancestry. And so this is really surprising to us because you'll notice that there are no geographically proximate desert bighorn herds anywhere close to this. So we're not really sure what happened here. And this is a really exciting and interesting result that, yeah, we weren't looking for. And so the last question we had was about the genetic diversity among different lineages. And so on the x-axis here, we have the three different subspecies in Nevada, again, colored in the same exact way. And on the y-axis, we have a measure of genetic diversity. And you'll see that there's really no apparent difference in genetic diversity across these lineages, which is reassuring. It shows us that, I don't really think that we're losing a whole lot of genetic diversity following these translocations. Now, to sum up uh, what we found so far, thinking about those genetic consequences, it doesn't appear that we're losing genetic structure among uh, these different populations. There's still strong genetic structure. Um, we do have some evidence of hybridization between Rocky Mountain and desert bighorn sheep. And Endow has done a really great job at maintaining levels of genetic diversity in all of these different lineages. And so it looks like, aside from the hybridization, there haven't necessarily been that many negative genetic consequences in the last 50 years of translocations, which is really, really awesome. So yeah, that's a great, I don't know, an exciting thing that I was able to tell Mike, and he's really excited. So, for the remainder of my time, I just want to tell you a little bit about what I'm doing moving forward. Uh, it's mostly with California bighorn sheep, but hopefully you'll find it interesting. And so, most of my focus moving forward will be thinking about the future management of California bighorns using genetics to help inform that. So back in 2000, Weyhausen and Ramey did a study on the cranial morphology of Rocky Mountain versus California bighorn sheep, and they found that they overlapped a lot in morphology space, suggesting that maybe Rocky Mountain and California bighorn sheep aren't that different from one another. This led some jurisdictions to start mixing these two lineages together in order to raise levels of genetic diversity. But from our study, we can see that Rocky Mountain and California bighorn, at least those populations in Nevada, are strongly genetically differentiated. And so that might lead us to wonder if we should actually be mixing these um, lineages together via translocation. Now, the reason why this is such a big issue is that most California bighorn populations found in the United States were all founded from serial translocations and sourced from only a handful of populations in British Columbia. And these founder effects could result in really low levels of genetic diversity. We've seen this before in studies of Oregon. And translocations of Rocky Mountain bighorn into North Dakota, where California bighorn were previously, yielded higher recruitment. So there might be some benefit of moving Rocky Mountain bighorn into these populations. And so really our main question is, are there circumstances when we should be mixing California and Rocky Mountain bighorn together? And to do that, we've started generating a range-wide and genome-wide genetic data set across, from British Columbia down to Colorado and thinking about sampling native herds and reintroduced herds to really be able to characterize the degree of separation between those two lineages across the range, and also to determine if there's thresholds of genetic diversity where we should start thinking about intervention via translocation. And so this really has been spearheaded by Clint Epps at Oregon State University, and again, Marjorie Matoke, myself, and Tom Parchman at the University of Nevada, Reno, and members of the Wild Sheep Genomics Working Group. But really, there's so many more people that have contributed, and we have collaborators at universities and state and federal agencies across Western North America. And none of this would be possible without all of the folks on this. I'm really excited to be able to, lean on everyone that's interested in collaborating. And we're about to send off the final samples to the sequencer now. And so hopefully fairly soon, I'll be able to tell you a little bit more about the genetics of these two lineages. And so I'd like to end by thanking my funding sources. Most of this has been funded by, or I guess entirely funded by the Nevada Department of Wildlife and the Wild Sheep Foundation. And if you have any questions, please let me know. Thank you so much for your time. I really appreciate it.